Hello and welcome to my channel. In this lecture we'll start talking about the cranial nerve nuclei, so let's begin. First of all, a general idea about the modal classification of neurons, that is to say the cranial nerves and any nerve all over the body, by the way. We have sensory or afferent sensations and motor or efferent functions. And for the sensory, they are divided into general and special types or subtypes. The general sensory, we have somatic sensory, means nerves that bring sensation from the skin and muscle. It's called general somatic sensory or general somatic afferent, GSS or GSA. Also visceral sensory afferents coming from the viscera, sensations from the viscera. This is called general visceral sensory. Special types, we have the somatic sensations coming from through vision and hearing. The vision and hearing are classified as somatic. And the sensations coming through taste and smell are classified as visceral sensory from taste and smell. For the motor functions, by the way, you will not find any sensation outside these four categories. For motor, it's the same: general motor, general somatic motor, or efferent to somites all over the body. And in the head, the muscles derived from somites as well. And general visceral motor, this is the parasympathetic or the autonomic to the viscera. It's called GVE or GV motor. Special type of motor to muscles of branchiomeric origin. That's these are the muscles derived from the pharyngeal arches, and it's called special visceral motor. There is no special sensory special somatic motor here. There is no special somatic motor. So we have 7 subcategories of sensory and motor modalities. This is a table I chose for you to that indicates the types of fibers in each nerve, starting from the oculomotor, which is the third cranial nerve, through the hypoglossal. And as you can see here, the somatic nerves that contain somatic motor, oculomotor, trochlear, and abducens, spinal accessory, and hypoglossal, and so forth. You can study this table and take your time studying it. But it's important to know that nerves, the cranial nerves that carry the autonomic, which is the parasympathetic secretomotor fibers, are 3, 7, 9, and 10. 3, 7, 9, and 10. Remember that the muscles derived from pharyngeal arches are supplied by nerves 5, 7, 9, and 10. That's trigeminal. But here, if you want to learn these by heart, the nerves that carry autonomic or parasympathetic are 3, 7 facial, 9 glossopharyngeal, and 10 as the vagus. The dorsal and ventral horns of the spinal cord, they develop from structures called the Ailer Plates and basal plates. These are the basal plates and these are the Ailer Plates. They are the same in the brainstem, but will be displaced because of the growth of the fourth ventricle, keeping the relation between the sensory, motor, and autonomic areas of gray matters maintained their relationship relative to each other. Sensory are usually lateral, as you can see here. Sensory in the brainstem lie lateral to the motor areas, and there is a corresponding intermediate area that serves as general visceral functions or means autonomic functions, as you can see here. These gray matter nuclei form a series of columns that extend through the brainstem, and extending from the gray horns of the spinal cord. You can consider the column as an upward extension of its corresponding area in the spinal cord, and the column in line with the ventral horn contains nuclei where the cell bodies of somatic motor neurons are located in the ventral horn, and the same in the somatic sense. In the sensory areas, they contain sensory neurons that are related to the ones in the spinal cord, especially those for pain and temperature, for example. Extension of the dorsal horn substantia gelatinosa of Rolando. You studied this with the spinal cord before. There are 6 columns of cranial nerve nuclei to be identified along the length of the brainstem. Column 1 is the most lateral, contains somatic sensory nuclei. Column 2 contains special visceral sensory, like the taste, for example. Column 3, general visceral sensory. Column 4, general visceral motor, which is the autonomic. See, the autonomic is in between the sensory and the motor. And then column 5, special visceral motor. These are for muscles of branchiomeric origin. And the somatic motor is the most medial column, and it carries motor impulses to muscles derived from somites. Now, the position and number of cranial nerve nuclei for each cranial nerve, not considering those serving as special senses, you can use some rules for working out the cranial nerve nuclei for nerves. 3 to 7 and 9 to 12 rules are as follows. There is one nucleus for each function of the cranial nerve on each side. Sensory nuclei lie lateral to motor nuclei, as we studied in the relationship in the brainstem before, and the nuclei that are at level at which the cranial nerves attach to the brainstem. So you'll expect to find the nuclei of the cranial nerve at the same level it leaves the or attaches to brainstem. As you can see here, the brainstem is divided into 2 halves. This showing the sensory nuclei. This is the trigeminal nucleus, for example, the mesencephalic, the main sensory nucleus, and the spinal nucleus and tract of the trigeminal nerves. And on the other side, you will find the motor nuclei, like the oculomotor, trochlear. This is the autonomic nucleus of the oculomotor, which is the Edinger-Westphal nucleus, and so forth. Of course, you have sensory on both sides and motor on both sides. Now, starting with the first cranial nerve, which is the olfactory nerve, it's unique that it doesn't have a thalamic relay before reaching the cortex. We used to, for all sensations coming to the cortex, they must relay in the thalamus first, thalamic nuclei first, but this special sense doesn't have a thalamic relay before reaching the cortex. And there are 3 types of cells or receptors, olfactory receptors. Basal stem cells, these are cells for regeneration. Support cells, called sustentacular cells, and olfactory receptor neurons. These are the olfactory receptor neurons you can see here. Now, they have unmyelinated axons that pass through the cribriform plate of the ethmoid to form the olfactory bulb. This is the olfactory bulb. And axons synapse with cells called the mitral cells in the form of glomeruli, as you can see here, of the olfactory bulb. Axons form the olfactory tract on its way to the olfactory cortex after dividing into medial and lateral stria. This is the olfactory tract. As you can see here, it's dividing into medial stria and lateral stria. See, lateral stria, medial stria. The medial stria will go to the septal area and the areas around it, and the lateral stria will go to the uncus and the olfactory cortex, which is called the entorhinal cortex. This cortex is called the entorhinal cortex. I told you before, as mentioned before, some axons of the medial stria reach the septal area. Other axons cross as the anterior commissure. I'll show you a picture of the anterior commissure. And these function as an inhibition to the contralateral mitral cells to be able to localize the olfactory stimulant. Axons of the lateral stria terminate in the pyriform cortex or primary olfactory cortex of the uncus and the amygdala, and what's so-called the medial forebrain bundle connects the olfactory cortex with the hypothalamus and brainstem to regulate autonomic responses like arousal through the reticular formation, salivation when thinking of food or watching food, and gastric contractions when watching or smelling food being prepared. This is the coronal section of the brain showing you to the anterior commissure. This is the anterior commissure, see? This is the caudate nucleus, the lentiform nucleus, and the internal capsule descending. Now, to clinical test the olfactory nerves, to know the function, by holding a strong smelling substance, such as smelling salts, under each nostril in turn while occluding the other nostril. This is how to test for olfaction. For the second cranial nerve, which is the optic nerve, the first order neurons are the bipolar cells. See, these are the bipolar cells. And the second order neurons are ganglion cells. These are ganglion cells. So these are bipolar cells. These are ganglion cells. And these are the photoreceptors, which are the rods and cones, as you can see here. Rods and cones. We have a bunch of cells that regulate the outflow, like the horizontal cells help integrate and regulate input from multiple photoreceptors, as you can see here, and the amacrine or amacrine cells that regulate the outflow of the bipolar cells. And the axons of ganglion cells are the ones that form the optic nerve. This is the inside of the retina. This is the outside of the retina. This is towards the vitreous humor. This is towards the outside. So the light comes and penetrate all these layers to hit the layer of rods and cones, and the impulse goes back in the opposite direction to be transmitted through the optic nerve again in the opposite direction. So light, impulse, electric impulse, and to the outside in the opposite direction. The ganglion cells transmit visual information from the retina to the several regions of the cortex, the occipital cortex. We studied this before. This is a cut section of the retina showing the area of the fovea where the maximum density of cones is there, and there are no rods in this area, for this is the area of highest visual acuity. Although rods are more numerous than cones, 120 rods for every 7 million cones. Now, rods are more sensitive to light. Don't confuse that they must be in the fovea. No. Rods are more sensitive to light, means light and dark. More sensitive than cones. But they are not sensitive to color. Okay? Cones are responsible for the high resolution vision. That's why it's concentrated in the fovea. And three types of color sensitive cones. Those are sensitive to red waves and green waves and blue light waves. The central visual pathway, axons of ganglion cells, as we saw before, form the optic nerve, coalesce to form the optic nerve at the optic disc, and then leaves the eye. As it leaves the eye, the optic nerve acquires a myelin sheath and meningeal coverings. That's why you can detect increased intracranial pressure by examining the retina. You will see a papilledema, and if there is a case of increased pressure in the cerebrospinal fluid, or the CSF, inside the skull. And anterior to the infundibular stalk, the two nerves, which means the optic nerves, form a chiasm. As you can see here, only the nasal fibers coming from the nasal retina that cross, see that? And the temporal fibers coming from temporal retina remain at the same side. So the chiasma is formed by two types, by the two nasal retinae of both eyes. Then the optic tract is formed and courses around the midbrain on its way to the lateral geniculate nucleus in the posterior aspect of the diencephalon, and they give branches to the pretectal area and superior colliculus to mediate pupillary light reflex. You can study this in further details in the vision lectures in the general anatomy. The lateral geniculate nucleus is the principal subcortical site for processing visual information, and inputs from, for example, the right hemiretina of each eye project to different layers of the right lateral geniculate nucleus to create, that is, from the temporal and from the nasal part, to create a complete representation of the left visual field. Right eye, left visual field. Right half of each eye is the left visual field, to be precise. It's composed of 6 layers. Layer 1 and 2, called the magnocellular layers. Layers 3 through 6 comprise the parvocellular layers. All these layers project to area 17 as the optic radiation, which divides into upper parts, upper and lower fibrous parts. Axons leaving the lateral geniculate nucleus form the optic radiation, as mentioned before. It continues to the primary visual cortex and divides into lower portion and upper portion. Fibers in the lower portion of the optic radiation, be careful here, the lower portion of optic radiation, not the lower portion of the field. Lower portion of optic radiation means the upper part of the field. Okay? So the lower portion of the optic radiation terminates in the lower half of the primary visual cortex. From the upper portions, which means the lower field, terminate on the upper half of the cortex. So the image is inverted in both vertical and lateral dimensions, and the primary visual cortex receives information from the contralateral half of the visual field. Some lesions in the visual pathway, see these lesions. This is the optic nerve, optic chiasma, optic tract, optic radiation. This is the inferior bundle and this is the superior bundle. In lesion number 1, where there is complete division of the optic nerve, as you can see, the right eye here is completely blind and the left eye is healthy. In the case of chiasma, like in pituitary tumors or craniopharyngioma, for example, pressing on the chiasma, there will be interruption of the nasal retinae. Fibers coming from nasal retinae means the lateral field, so it will be bitemporal hemianopia. It's called bitemporal hemianopia. In the optic tract at one side, so it has the temporal retina of one side and the nasal retina of the other side. So the lesion will be directed in the same direction, like here. This is homonymous hemianopia. In this case, in the right eye, the left field, and the left eye, the left field also as well. In lesion number 4, it will produce, after dividing into upper and lower portions, the optic tract. Because this is the lower portion, so it's the upper field, it will produce upper quadrantanopia. And the difference is with the lesions proximal to the occipital cortex that there will be quadrantanopia, but with macular sparing. The macula will be spared. These are some lesions of the visual pathway. I hope you got the maximum benefit from this lecture, and thank you very much for watching.